Hello everybody. In this video I want to go over mitosis. We're going to talk about the stages of it, what happens in each stage, and how those actually allow the chromosomes in the nucleus to divide evenly. So if you don't know what mitosis is or where it fits in the cell cycle, you may want to go back and watch the video that I did over the cell cycle and the stages of that, so you get an idea or an understanding of really how this plays into the normal life cycle or stages that a cell goes through. But just to recap, mitosis itself is the division of the nucleus and its contents. And so when we talk about mitosis, a lot of times you'll hear it referred to as a cell division process, but ultimately what mitosis is dividing the nucleus and specifically the chromosomes inside that nucleus. I took this PowerPoint from a colleague of mine, a friend of mine, and I don't know where they found this animation, but it kind of shows you the stages of mitosis and what's going on there. So mitosis itself actually has 4 distinct steps to it, and those sometimes get broken down even further. I'm just going to do a real quick basic overview of those 4. And to remember those stages, we use an acronym, PMAT. And so this will help you remember the 4 stages or phases of mitosis and the order that they happen. That first one is, the P stands for prophase, and so prophase is the first one, and that's actually, if we look at the animation, what's going on right now. And at the end of prophase, we have metaphase, which is going on now in the animation, and then anaphase, where the chromosomes split apart and move to opposite sides, and then we end with telophase. So if we go back to the animation, here's metaphase and then anaphase, and telophase happens right now. Cytokinesis is another stage of the cell cycle. It's separate from mitosis, and so going back, we have the interphase part of the cell cycle with gap 1, synthesis, and gap 2. At the end of that, we have mitosis take place, or the mitotic stage, sometimes that's referred to. And at the end of mitosis is cytokinesis. And if we break this word apart, cytokinesis, cyto means cell, and then kinesis is talking about movement. And so cytokinesis is really the movement of all the things inside the cell, or ultimately cell division. So I do want you to know that these 4 phases of mitosis are different and distinct, separate from cytokinesis. So they're 2 different things. Well, let's dive in and take a look at mitosis in a little more detail. Like I was saying, this happens at the end of the cell cycle as the cell is getting ready to divide or getting ready to go through cytokinesis. Going back into the cell cycle with just a little overview, in a normal human cell, during interphase, during really gap 1, that first stage of the cell cycle, there's 46 chromosomes in the nucleus of the cell, and all 46 of those are important for that cell to be alive. There's 23 kinds of chromosomes, and we have 2 of each kind of chromosome. We get 23 of those, or 1 set from mom, 1 set from dad. And I went over that at a little bit of detail in the chromosome structure video, so if you want, you can go back and watch that. But during the S stage, or the synthesis stage of the cell cycle, those chromosomes get copied, or DNA is replicated. And we go from 46 to 92 total chromosomes in there, because the chromosomes are duplicated. What I find really interesting about the stages of mitosis, this whole PMAT cycle, is that those chromosomes are able to divide evenly into 2 new nuclei, and so that each new nucleus that's formed has the right 46 chromosomes that it needs to function. It's not just taking 92 things and separating them into 2 even groups. It actually has the right 23 pairs of chromosomes so that those cells can function. If something messes up there, if that doesn't happen correctly, then the new cells that are formed are going to die. They just don't have the right genes and the right chromosomes to be able to survive. And then cytokinesis happens, and we end up with 2 daughter cells that are genetically identical. They have the same 46 chromosomes in them. So let's look at each of these stages in detail and see how this actually happens. And so here we see P, or prophase, the prophase stage, metaphase, anaphase, and telophase here. So I want to go into detail about what's going on in each. You can write that down or take notes or references however you want to on this. But in our prophase stage or phase of mitosis, there's a lot going on here that I really want you guys to see. During prophase, our chromosomes, or our DNA, actually condenses and coils into these really tightly packed chromosome structures that you guys are probably familiar with, the X-shaped things. Going back to the chromosome structure, remember the two sister chromatids are genetically identical. They're made during DNA replication, so these are the same genes, the exact same DNA in both of these sister chromatids. Those are held together in the centromere region. There's a protein clump referred to as a kinetochore that holds all this together. And so our chromosomes become visible. They start to form. This little dotted line you see here, this represents our nuclear membrane or our nuclear envelope. During prophase, that starts to dissolve. There are some enzymes that are made during GAP2 that actually start to break that envelope down or that membrane down. So our chromosomes then become exposed. The other things that happen during prophase, our centromeres here, centrioles, I can't getting words mixed up. Our centrioles are these organelles that produce these proteins called spindle fibers. And the spindle begins to grow from these centrioles. As that happens, the spindle fibers will come in contact with each other here in the center. Well, they actually repel each other. They push each other apart, kind of like poles on a magnet. If we have the north and the north pole of two magnets together, it pushes them apart. Well, the centrioles are still producing spindle fibers down here, so these are getting longer, but they're pushing each other apart. And what that does is it moves the centrioles to opposite ends of the cell. So all that happens in prophase. Chromosomes become visible, the nuclear envelope or nuclear membrane dissolves, and our spindle fibers start to form. During metaphase, those spindle fibers, some of those reach out and will actually attach to that kinetochore protein in the centromere region of our chromosomes. When they do, as these spindle fibers grow and push, it actually lines these chromosomes up nice and neat at the metaphase plate of our cell. Some of the spindle fibers don't actually touch chromosomes, and they keep repelling each other. And by doing that, they push the centrioles further and further apart, which is spreading the cell out, growing it a little bit. But the ones that are attached to that kinetochore line these up, and this is incredibly important. If you notice, we have these two sister chromatids here. Remember, they're identical copies of each other, and they're lined up nice and neat. And so in metaphase, we line those up. But then in anaphase, the centromere, or the kinetochore proteins there at the centromere split, so the sister chromatids are separated from each other. And so this copy goes to this side of the cell, this copy goes to this side of the cell, and it separates those nice and evenly. And so in this case, I'm going to end up with 4 chromosomes on this side and 4 chromosomes on this side, and they are going to be the exact same chromosomes, and each of the 2 nuclei that are formed will be genetically identical. So in anaphase, the sister chromatids are split at the centromere region and move to opposite sides of the cell. In telophase, our new nuclear membrane or new nuclear envelope will form around the chromosomes at each side of the cell, so we end up with 2 identical nuclei. The spindle fibers start to disappear, the chromosomes kind of uncoil a little bit, and cytokinesis begins. And so really quick, in prophase, chromosomes are visible, spindles forming, nuclear membrane dissolves. Metaphase, the chromosomes line up at the metaphase plate. Anaphase, the sister chromatids are split at the centromere, and in telophase, 2 new nuclei are formed. In this case, there was 4 chromosomes in this cell, but in a human cell, remember, we have a total of 46, or really 23 pairs, but a total of 46 chromosomes that would line up. So when we're dealing with that large number of chromosomes, it's really important that they line up this way so that the right 46 chromosomes go to the opposite sides of the cell. Here's just a picture of that through of some animal cells in a microscope. We can see an interphase. Chromosomes aren't really visible. Here they do become visible and they line up in metaphase. In anaphase, they're starting to be pulled apart. We can see the spindle fibers reaching across here and grabbing hold of the sister chromatids. And in early telophase, I can see that they've moved to the opposite sides of the cell. And in late telophase, I've got the 2 nuclei, and I can actually see that this cell is starting to divide and cytokinesis is taking place. So that's just showing you some actual microscope viewing of the cell division taking place. So take a look at this, pause the video really quickly, and see if you can identify which stage of mitosis each one of these cells is going in. Once you think you've got it, go ahead and push play, but I'm going to give you the answers now. So if you were wondering, let's start with number 1 here. This right here, we can see the chromosomes visible. Those are there. That's going to be anaphase. Number 2, they're lined up in the middle. That is our metaphase stage. Number 3, we have prophase. I can see the chromosomes condensing there. Number 4, this one here is going to be telophase because I've got the 2 different nuclei forming. And 5 right here is interphase. This is the cell just going through its normal day-to-day routines. Hopefully that clears up mitosis, kind of what happens in each stage, and it gives you a little bit of detail about how those chromosomes are split evenly during this process.